The next application of biotechnology in agriculture specifically is in the production of the pest-resistant plants in tobacco, specifically against nematode resistance. Now, what is this nematode resistance? There are several nematodes which which act as parasites on a wide variety of plants and animals, including human beings. Nematodes are parasites of even humans. One such nematode is Melodygium incognita. Please remember the name, again very important for your objective. So this Melodygium incognita, it attacks the roots of the tobacco plant. So its host, it's a parasite that whose host is the tobacco plant roots, and they cause a great reduction in the yield of the in the entire tobacco crop. Now, why they cause this disease, which is called as root knot disease? See, tobacco is a taproot system. It's a dicot plant. It's got a taproot system, and look at that root. That root becomes completely useless after some time. There is what is called as those tumors, those tumors that are produced. This in fact it is also called as the crown gall disease also. Okay, so this is called these root knots or root tumors it will create, and it will destroy the plant's life. And this is the worm Melodygium incognita. If you can see, this is the root cells, and these are the worms that are present, the parasites, the nematodes which are present in the root. This is the root, the knot of the root. Okay. Now, this Melodygium incognita, it can be, you can protect the tobacco plant against it by using RNA interference. So, what is this RNA interference? RNA interference is in short, it takes place in all eukaryotic organisms. It's not nothing new. It takes place in all eukaryotic organisms, but at the cellular level, not at the organ or the tissue level. This happens at the cell level, and that's why it is a cellular defense mechanism. When we talk about organ level defense mechanism, you have the immune system, but at the cell level also you will have certain defense mechanism which is created by RNA interference, or it is also called as PTGS. It is also called as PTGS, means post transcriptional gene silencing. Post transcriptional gene silencing. So you basically, after the formation of RNA, post transcription is what? When the mRNA is formed. Now, even after forming the mRNA, if you are able to somehow make sure that this mRNA is not functional, then you are silencing the gene. Gene is silent, that means there is no protein coming from that gene. Gene is expressive, that means you're getting some sort of protein or whatever product that gene is supposed to give, you're getting it. So if a gene is expressing, that means it underwent transcription and translation. But if after transcription you're stopping the mRNA, you're not allowing the mRNA to work, then there will be no translation, and hence you will silence the gene. So this kind of defense mechanism is mediated by the cells, and it is present in all eukaryotic organisms. And this is used as a tool to prevent the nematode effect, or prevent the harmful effects of nematodes in the tobacco plant. And I've said basically what it does is you take a specific mRNA and you silence that mRNA by preventing the translation. How can you do it? We know that mRNAs are single stranded. Okay, so if you find another RNA which is complementary to it, okay, then you have the double-stranded mRNA, and this double-stranded mRNA cannot undergo translation. It will fail in translation because RNA has to be single-stranded, then only it will undergo translation. Remember, we have discussed all of this before, transcription and translation. So that's in this way you're creating gene silencing. How? By making specific mRNA into a double strand and preventing the process of translation. So RNA interference basically uses agrobacterium vectors. Agrobacterium vectors, as we remember in the previous chapter, agrobacterium vectors are basically the TI plasmids that are taken from the Agrobacterium tumefaciens bacteria. So they are the TI plasmid vectors which are taken from the agrobacterium, hence they are called as agrobacterium vectors. And we've also discussed that for plants, for creating biotechnology work in plants, TI plasmids vectors are very useful. So through this plasmid, you are introducing nematode specific genes that are specific for 𝐵 incognita. Where are you introducing? In the host plant. That means you are introducing it in the cells of the tobacco plant. So you are directly putting the 𝐵 incognita genes in your tobacco plant. Okay, so this is the TI plasmid, and you are taking the TI plasmid and you are adding that to the TI plasmid, the nematode specific genes. Okay, so now what happens? Once the nematode specific genes are introduced into the plant cell, this is the plant cell. So this plant cell already contains the nematode specific genes and it produces a double-stranded RNA. Sometimes either you introduce that or you directly allow double-stranded RNA of the nematode genes produced from outside, or you will give it in the plant cell. You do two things. Either you give the genes directly in the cell, or you give the double-stranded RNA for those genes by transcribing it outside and putting it into the cell. Once there, the double-stranded RNA, it will be processed by an enzyme called dicer enzyme. This dicer also has a name called RNase 3. So this dicer, what it does is it takes this double-stranded DNA, binds to it and breaks it into small pieces. These small pieces are called short interfering RNA. Short interfering RNA or siRNAs, and these siRNAs are 21 to 23 nucleotides long and they will be in the cell. So they will be floating in the plant cell. Okay, great. Now you have the nematode. When the nematode 𝐵 incognita attacks the root of a plant or root cell of a plant and tries to eat it, this short interfering RNAs enter in the nematode cell. The siRNAs are then taken up by the nematode, where they enter into the cells of the nematode, and there these siRNAs, they form a complex with they form a complex, or they form a bond with RISC. Now what is RISC? RISC is RNA induced silencing complex. Okay, so you have, this is the RNA introduced silencing complex, and to it your siRNA is bound. The short interfering RNA, they are double stranded. Once they bind to the RISC, they will become single-stranded. And these single-stranded RISCs will bind to the mRNA that is specific for the nematode. That is very important for the nematode survival. Okay, and by binding that, they will cleave, they will destroy that mRNA, silencing the nematode genes. Without the major proteins, because those genes were silenced, the nematode cannot survive and it will die. So this is nematode specific pest resistance created by RNA interference. So in the same way, various transgenic plants have been produced, all of them which are used in agriculture. Transgenic plants are nothing but genetically modified plants or genetically modified plants in which there is a foreign gene. A foreign gene is transferred into the plant cells. Okay, so plants with desirable characters created through gene transfer, that is called as your transgenic plant. So let us say this is the genome of my plant. Okay, and this is a foreign gene which is which can be taken from another plant or a completely different organism or a bacteria, anything. Now I will transfer this gene into the plant cell, and now I will allow this plant cell to grow and develop, and that is my genetically modified plant or my transgenic plant. Uh, Basically what biotechnology says, transgenic means specifically you're transferring gene from outside to into a plant cell. Whereas in genetically modified plants, there is a case that you don't transfer anything from outside, but you can modify what is there in between. You're understanding it? Transgenic plants specifically means you are taking a foreign gene from outside and putting it into your plant. So there is definite transfer of a gene. But all genetically modified plants are not transgenics. Why? Few genetical modifications happen within the genome. There is no gene transfer. Understood? So specifically transgenic plants means you're taking genes from outside. Okay, so like this is the methods of transgenics. First, you identify the gene. Whatever the gene of interest is, you will take that gene. Then you will put it in the plant cell. This is my plant cell. Then you allow the plant cell to grow using tissue culture. In that you select the best transgenics and grow these genetically modified crops or transgenics in a greenhouse and then release it in the commercial market. Okay, now though a number of methods have been developed to introduce the cloned genes into the plant cells, the best effective vector is TI plasmid. Remember, TI plasmid is the most effective vector for transgenics in plants. Now, several transgenic plants have been produced to meet specific needs of agriculture and human life. We have already seen transgenic tobacco, pest-resistant tobacco. You've also seen BT cotton. Then you have plants, transgenic plants that have been designed to produce post-harvest loss. Okay, then you have the transgenic tomato, which is bruise resistant. Now, this is an example of a plant which is a transgenic that is designed to reduce post-harvest loss. Now, what is this post-harvest loss? Normally, we all have plants which they are produced in various parts of the country and they are transported to various parts of the country. For example, let's take the case of mango. During the mango season, mangoes do not grow everywhere in India. In few parts of India, they are grown, they are harvested, then they are transported through various plants. And during this transport, there will be loss because of rain, because of traffic jams, or because of getting injuries in the trucks and the containers. The fruits get destroyed. One such fruit that shows a most important post-harvest loss is tomato. Now, tomato, if you harvest a ripened tomato, okay, let us say it is produced in Andhra Pradesh. I have taken ripened red beautiful tomatoes. I will put it and I have to send it to Kerala. By the time the truck starts from Andhra Pradesh and goes to Kerala, it might get bruised, it might get rotten, and it might get spoiled. Okay, so what do people do? Agricultural scientists, they want to produce a tomato which is suitable for storage and transport and delay or ripening and offer shelf life. So what are the things here that they are doing? Suitable for storage, transport, delay ripening, longer shelf life. Now this is interesting. Delay ripening. Why is this? Normally when you want to produce, when you want to sell tomatoes, what the farmer does is he does not take a ripened tomato, he takes the green unripened tomato, okay, and he puts them in containers. He sprays ethylene gas or ethylene products on them, and as a result they hasten the ripening process, and it is done during the process of transportation. So when they remove the tomato and when they are starting the transport, it is a raw tomato. Either after reaching the destination or in between, they subject that tomatoes to ethylene and then they get ripened. So you're hasting the ripening process and you're doing it artificially. So the color of the tomato doesn't look good. I mean, we all hear tomatoes are supposed to be red in color, but if you ask your mummy, half of the tomatoes are orange in color for us in India. Why? Because early hastened ripening does not give proper color, the taste will not be good, there will be no proper nutritional qualities, all of them will not be available. But the farmers don't have any other choice. If they take a ripened tomato, what happens? It gets bruised and it gets destroyed. Even before it reaches the place, half of the tomatoes are either rotten or they're squished or they're damaged. So the only way for them is to traditionally send this. But if you take a flavor saver tomato, that tomato is modified in such a way that it is bruise resistant. Okay, you can store it for a longer time, it will not get destroyed, you can transport it for longer periods, it has got longer shelf life, means even after getting detached from the plant it will not rot quickly, and it slowly ripens. So if I take a tomato, flavor saver tomato from Tamil Nadu and I send it to Jammu and Kashmir or Ladakh, they will be absolutely fine, they are not destroyed, they are slowly ripened, they taste good. So that's the concept behind flavor saver tomato. Now, who produced this flavor saver tomato? See, normal tomato. So, normal tomato, this has a particular gene called the PG gene, which is polygalactourease. Now, this is an enzyme that digests the pectin. It breaks down the pectin of a raw, unripened fruit, and as a result, the cell wall becomes more susceptible to rotting and damage. So, that is in the normal tomato. Now, in a transgenic tomato, which is a flavor, don't look at this. In a traditional flavor saver tomato, you have the antisense RNA to the PG gene. So that the PG gene, the mRNA, the normal mRNA of the polygatlonears, instead of single stranded, it becomes double stranded. So this is a kind of RNA interference only, where instead of cleaving the RNA, you are producing the double strands, and as a result you are producing the complementary mRNA which is double stranded, and the polygalactourease is the enzyme quantity is completely reduced, and as a result the fruit stays ripened and there will be no rotting for about 3 weeks. Similarly, you have plants which have increased efficiency for mineral usage. This prevents the exhaustion of the fertility of the soil. There are tailor-made plants which in the case of for the production of fuels. Okay, then there is high yielding and disease resistant variety plants like that of the transgenic papaya, which is resistant to papaya ring spot virus. Okay, and there is this transgenic tomato which is resistant to the bacterial pathogen Pseudomonas. Transgenic potato which is resistant to the fungi Phytophthora, and transgenic golden rice which is the Taipei form which is enriched with vitamin A to prevent blindness. Then hybrid street production, where you have male plants of this Brassica napus which are completely sterile, and You also have the basmati variety of rice which is resistant against abiotic and biotic stresses like that of chemicals, cold, drought, salt, heat. You have plants that are resistant to only abiotic stress